Okay, then you tell me when and I start.
Yep, I said yes.
Yep. Okay, shall we start, Andrea?
Yeah. No, yeah, now. Go.
Then you tell me when and I start.
Now you can start.
No, okay. No. I will first introduce you. So today we have the pleasure to have with us Jean-Francois Rupprecht. He is a group leader researcher at the Centre de Physique Théorique in Marseille, in France, hosted by the Laboratory of Adhesion and Inflammation. Jean-Francois did his PhD at the Sorbonne University, where he worked on random search processes. Then he went on with 1 year project at the École Normale Supérieure in Paris. Sorry for my French. Where he worked on magnetotactic bacteria, a fascinating class of microorganisms which can orient their swimming direction along the Earth magnetic field lines. Around that time, then in 2015, he had a great chance to meet with Prof. Jacques Prost. Actually, maybe we should invite him in one of our next sessions. And together with Jacques, Jean-Francois embarked on a postdoctoral stay at the Mechanobiology Institute in Singapore, where obviously is the place where we got to know each other, and where he worked on a project of developing a fluctuating active gel theory. So active gel theory had been successfully applied to several systems, ranging from cell division to embryogenesis, and indeed they worked in parallel on a variety of subjects, ranging from molecular motors to tissue mechanics. After this experience in Singapore, Jean-Francois has been recruited by the CNRS, so the French National Research Institute, in 2018. And thanks to a grant from the Turing Center for Interdisciplinary Research, the Centuri, in Marseille, he started the Out of Equilibrium Mechanics team, or OM, as he likes to call him, for Olympique Marseille, for soccer fans. And there, his group is actually having exciting process that are waiting for postdoc applications starting next year. So someone of you are interested after the talk, you can, think of sending an email to him. So now I'm looking forward to hear about this Escher's convex and concave epithelial tissue flows in curved environments. So thanks once again, Jean-Francois, and Jean-Francois, the floor is yours.
Thanks a lot, Christina, for this introduction, and thanks also to Andrea for inviting me. So it's a pleasure to talk today about convex problems, concave problems, and it's a bit of pretext to talk about different things, which I hope to find some unifying features along them. So here is a full painting, and what I will talk about today is about obviously convex and concave geometries on which we'll place epithelial tissue. I'm a theoretician. I will discuss the question of hydrodynamic theories in those geometries. In the second part of my talk, I will discuss cell-based models, which are called vertex models, where we found the emergence of a cellular pattern, which we could call the Escher phase, because it's one of the very regular feature, one of the regular motifs that Escher is using in his paintings, and you see one of them on those flags. In the last part of my talk, this one is, you see a tree here, and we do work on trees, neuronal tree architecture, which are segmented and tracked live. These are things we are developing with a PhD student and with data that comes from Thomas Lecuy lab. I'll be very quick on that part, but there's lots of statistical physics problems to be explored in those systems. And this is like a very emerging field of tracking the dynamics of these neurons during embryonic development. So I'll go back to the first part of my talk. Most of my talk will deal with epithelial tissue and the question of flows and forces. So I will start with a pretty long introduction on those subjects. So I've had the chance to work with Benoît Ladoux on experiments where they observed that when you place these MDCK epithelial tissues, on a soft gel, they can, whoops, sorry for that. Why is it not playing? This is like the universal law of, why is a movie not working? Ah, sorry, here it is. Of epithelial fracture, spontaneous fractures in MDC case. So here you see one of those holes, another here and another there. This happens for these flat epithelial tissues placed on a soft substrate. You do not see these holes on a more rigid substrate. I will not really discuss this aspect today. I will just have one slide on that, but I would be very interested in discussing about these problems too, and this is on bioarchive. But what I wanted to say with this movie is, if you look at it from a physics point of view, what can you say about it? What do you see? You see cells that are moving along, streaming, and one of the first things you can do is to track their velocity. So, I mean, this movie is a bit dirty. I'm sorry for the quality. Shuffling around this movie, but you can track the velocity. This is the first thing that people have done on epithelial tissue. You use PIV and stuff like that. What do you get? You get a polar field, a field of vectors, of velocity fields. This is the first thing you can extract from your movie. The second thing you can extract is cell shapes. And you can define an axis of elongation of your cells, which I'm representing here in red. This would correspond to the main axis of elongation. In fact, if you really want to talk about the cell shape, you should define a tensor with a major axis, a minor axis, and an orientation. Here, for convenience, I just represent the major axis of elongation. And this you can extract from your movies in several ways. Many people have been using orientation G, which is a software in Fiji, to extract that, and that's what happened in that movie. You can also perform segmentation and extract the cell shape. These are things we are currently doing with a PhD student in the team. And actually, with recent tools such as CellPose or deep learning codes like that, you can actually get very good quantitative measures of the cell shape. But this is another story. What can you extract? What can we measure in experiments? First thing, cell velocities, PIV methods, cell shapes, whatever the method. And there's another thing that you could extract by using soft gels with beads into it. And Andrea is familiar with that because we've discussed on that for some time. It's called traction force microscopy. You can infer from the bead displacement on top of which the tissue is. From the bead displacement, you can extract maps of stresses within the tissue. And this is what you can show here, an evolution in the map of the local pressure field. And so here again, a stress is a tensor. It's got four independent components. And here I'm just representing the pressure, the trace of that matrix, 2x2 matrix. But what you see here is that during the movie, as the cells move, deform, you have fluctuations in the stresses experienced by the cell. So the cells are pushing each other, pulling on each other, and this is measurable using this traction force microscopy. So if you think from the eye of a physicist, you have everything you want to create a hydrodynamic theory. A hydrodynamic theory is going to give you a relation between the cell shape, the map of velocities, and the stresses. And one such theory, which has been used over the last 20 years and which I will discuss in the next slide, is called active gel theory. The coefficients relating the stress and gradients of velocity is typically called a viscosity. This is called a transport coefficient. But because you have a local consumption of energy, you should also expect a coefficient, which we know zeta, contractility coefficient, which relates the stresses that you measure in your tissue to the local cell shape order, the direction of, essentially, the direction here in red. And what people have been using, there's been a paper in 2017 by Benoît Ladoux, who was also in the Mechanobiology Institute at that time, which correlated the cell shape and specific patterns in cell shapes called topological defects to regions where you have a higher stress. And what they found later is that it indeed trigger cell division, not cell division, sorry, cell death, also cell division, cell death and exclusion. So here, I would like to talk a bit about the definition of those particular patterns in cell shapes, which are like comet-like. These are called +1/2 topological defects for a reason that if you take a cell here and you define a vector along it, and you make it rotate around that pattern. So this pattern looks like this, okay? So it's really like a comet in the cellular orientation. So you take a vector along one of those lines of elongation, and you make it turn around the defect. So make it turn. So I have an animation, in case you haven't seen it on the left. You make it turn again, and you make it come back to the initial position here. And you've gained a phase. So you've rotated along the way, but your vector is no longer in the same orientation as before. It has an opposite direction. So because you do 2 pi times a factor 1/2, you get a pi phase. So, and the pi phase is by how much you've rotated. Okay, so this is why you call that a +1/2. And this is also the way you've rotated. You've rotated counterclockwise. So this is called a +1/2 topological defect. There are also other defects which are called -1/2 topological defects. And these are the first ones you would see by considering an assembly of rods. All of these rods are just a cell elongation. So these plus and minus topological defects are the first patterns in cell shapes that you can expect to see in a tissue, and you do see lots of them. So now you turn back to your hydrodynamic theory, and you've written that in first linear order in gradients of B and the expression in the cell shape Q. And you assume a pattern in cell shape along these lines, which I'm representing here. Okay, so you imagine here that all the cells are rods. It's not necessarily the case for MDCK, but you imagine that they're getting deformed. So you imagine you have rods which are placed along these white lines. Now imagine that this zeta coefficient is positive. What will happen, in fact, is that these cells, microscopically, these cells are pushing on their neighbor. Okay? This will lead to a region of tension here at the tip of the topological defect. So the tip is getting stretched. The tip of the topological defect is getting stretched. Now imagine that you have the opposite. You know, I was a bit embarrassed because I was like, this doesn't work. I'm sorry for that. I have a typo in there. These things should have been in the opposite direction. And how can I do that? Oh, this is embarrassing. Okay, let me just suppress it and imagine that these arrows were in the opposite direction. Play from current side. I'm sorry for that. Now imagine that your cells are pushing, and this time pushing on the others. And so it's this sign of zeta. Now here you have like a lens.
Sorry, Jean-Francois, when you say the opposite direction, because it's pointing in both directions, you mean perpendicular?
But it means that this thing, I wished I knew, I think that's what happened, is that I wanted to, I'm going to use a shape option. But I mean, pedagogically, it's quite good, in fact, because you know, making mistake is the best way to check that where can I want this arrow
to if you just describe the direction.
Yeah, but the best way would be, can I draw it? I can draw it.
I think you have draw function there.
Yes.
You see all means.
I'm doing it. I'm doing it. Do you see it?
Okay, perfect. Yes.
My arrows, I want
okay.
my arrows to point like that.
No. Okay, so compression, yeah, okay, perfect.
Exactly. Okay, let me go back. I'm sorry, I'm going to erase everything and I'm going to play it back. Okay, I was not really intending to spend so much time on that, but thanks to that type of, oh no, come on. Okay, so this one is in the opposite direction.
Okay.
Here it's pushing on its neighbor, here it's pulling on its neighbor. And what you see, you have a clear prediction by the theory. At the tip, at the nose of your +1/2, this is so annoying that I'm going to put it back here. Oh, come on. Yes, sorry for that. Yes.
What is set or sit?
This
+0. Yeah.
This zeta parameter, this is a free parameter. I mean, this is a parameter that you that needs to be. I mean, that is going to. This is a characteristic parameter of your system, a bit like the elastic modulus or the viscosity.
Right.
This is something that you fix for, I mean, that you want to fix for your tissue under study. And this is going to be one of the biggest results of the Nature 2017 paper, is that it's fixing the sign of this parameter zeta. It's saying for this class of NDCK cells, the zeta is negative.
And what is Q, big Q?
Big Q is a cell shape. Big Q is a tensor. I mean, I've got extra slides on that, but another thing that I could have said about it is that it's a tensor, and the reason why you can define that this way is that, okay, so this is something I didn't intend to, so it's a tensor, which you can define this way. So if your cells are elongated, okay, way you can construct, it's like an inertia tensor. And an inertia tensor, you can extract, an inertia tensor is going to be a 2x2 matrix, okay, where you average over the neighbor, giving them a mass, and a 2x2 matrix can be diagonalized. You have two major axes, one which is a major axis for elongation, and the other is a minor axis of elongation.
All right.
You have an orientation. So Q is a tensor describing not so much the individual cell shape, but an average orientation of the cell shape. These theories are said to be coarse-grained. I mean, you average the local cell orientation. Typically, in the Benoît Ladoux papers, they average over a box size of 2 to 3 cells. So that to have something which is not completely stochastic and to have something a bit smooth. Because that's also what you see in your tissue. I mean, if one cell is elongated, it's also, and the neighbor are also a bit elongated. I mean, roughly. Yeah. So this is a coarse-grained parameter. And so what I wanted to say, but thanks for the question. Thanks a lot for the question. Indeed, I mean, this is the Q is critical for my talk. So I'm glad you asked it. So here you see that if you have a +1/2 defect, at the head of the +1/2 defect here, depending on the sign of this parameter zeta, you're either under tension or under compression. And this is one of the big things of this Nature paper in 2017, is that what they find is that it's under compression at the tip of the +1/2 defect, consistently with a zeta being negative, and that's consistent with an increase in cellular extrusion there. You know, it's like a soap. If you press on a soap, it's going to pull out from your hands. Okay, and this is a bit the same thing with these cells. They're under compression, they expel out. So there's still in the introduction, there's been another experiment, 2018, where the Silberson group in Institut Curie placed RP1 cells, epithelial cells, still in a flat substrate, a monolayer, on adherent stripes, and they observed that they perform a spontaneous shear in those stripes. So here there's no fibronectin in the middle. What they obtained by averaging their movies. They find that these cells, which are quite elongated, as you could see here, on average, they have an orientation, which is displayed here. This is the average orientation of the cells, of the RP1 cells. And if you average also the movies, you see that you have a spontaneous flow of cells, with cells streaming downwards on the left and streaming upward on the right. And you've got roughly 50-50 of proportion. Sometimes the cells on the left could be going up, etc. It doesn't need to be like that. I mean, there's a bit of chirality, but not a fantastic amount as far as I remember. Maybe there are some chirality, I think. But this is not the most important thing I want to discuss here. If you go back to your theory, this hydrodynamic theory that I just wrote, it's able to describe this kind of behavior by assuming that, again, these cells, and it's not universal, some of the cells have an opposite contractility, these cells behave as an extensile system. So what as flow occur, the thing is that these cells tilt, and look at what's happening here. You have more cells pointing downwards towards the left here than cells. If you take a portion here, you have more cells pointing downwards. Here you have 4 cells pointing downwards and only 3 cells pointing upward. Okay, if you look at the left-hand side to the middle of the stripe, 4 cells pointing downward, 3 cells pointing upward. So you do expect to have a flow of cells that corresponds to what you have in your experiment, provided that your cells individually push on their neighbor. And this is consistent with zeta being negative. So there's a whole formalism, hydrodynamic theory formalism, to solve this equation and in order to prove that rigorously. But physically, that's a key take-home message, is that in this system, indeed, you have cells that are extensile and pushing on their neighbor. So what I wanted to say, what I want to now say is that for my talk and convex and concave, is that all of what we discussed was obtained in flat 2D geometries, both on the theoretical level and on the experimental level. Flat epithelial monolayers. Yet epithelial tissues are often curved at the micron scales, and that there are plenty of examples of that. And here we'll discuss a recent paper that we submitted on arXiv together with Jacques Pro. So, for example, in the gut, you have a monolayer of cells that are placed along this structure that are called villi, which have a length of about 500 micron, which is quite large, but at the tip of those villi, typically, you start to have curvatures which are comparable to the cell size, in fact. These are not the only examples. Of course, you have that also in vitro experiments. So here are cells in a villi observed by microscopy. Also, Wang Zi in 2017 from Delphine Delacour at Benoît Ladoux, René Marc-Mege lab observed the spontaneous rotation of cells in tubes. So experimentally, these becomes accessible in vitro system. And quite recently, there's been 2 papers where groups have placed epithelial tissues on corrugated sheets, okay, like really like on a rooftop. You can have these corrugated sheets, so infinite, that also look a bit like what you would have in some regions of the gut. And what they look actually more specifically is the architecture. How do the cells pack in those systems? What we will be addressing in the rest of that part of the talk is more about the flows. How do cells start to emerge to flow in exactly that geometry? So the question I want to address is what could be the role of the curvature of the underlying substrate on the global tissue flow properties. And what I want to say is that when you look at these individual cells on this curved substrate, there are two types of tendencies. Some cells tend to orient along the major axis of the curvature. Some other cells would orient in the opposite direction. So you really have like a variety of behavior, but clearly in some cell types, cells would really prefer to elongate against the curvature gradient, and some would really prefer to orient so as to maximize the felt curvature. And this could be related to mechanical constraint on the nucleus. Microscopically, there are many things that can explain why a cell would orient specifically regarding to a sign of the curvature. Here, we will not address why this is so. Why would a cell prefer a specific curvature or the other? This is not of our concern. What we want to understand is how this preference is going to affect the collective flows of your system. A bit in the same way that in the previous introduction part, we did not really investigate at that stage why would cell push on their neighbor. The question was, if the cell push or pull on their neighbor, what is the resulting stress patterns? What are the resulting flows? Here we have the same spirit. This is the spirit of continuum theories and hydrodynamic theories. What we've included compared to the previous framework is curvature. Curvature affects the operators. You now have a metric, a bit like in general relativity, for example. I mean, you have to use differential geometry tools to redefine your Laplacian, your gradients, etc. This is one contribution. But there are also some other contributions that we just discussed, which are the preference of individual cells to orient in a specific direction with respect to the curvature. And this you can include in your free energy, defining the cellular orientation. In principle, you could also include a preference of the cells to orient their velocity in a specific direction of the curvature. This we did not include in our study, but it could also be something interesting. We really focused on the nematic aspect. So how do the cell shape, still the same parameter Q, how is the cell shape coupling to the curvature affecting the transition to flows in those systems? And this again can be related to stress fibers. So I will skip on the math, but it's doable. We have an external field coming from the curvature coupling, and it's using the same techniques. Adapting the techniques, we can solve for the flows. And here are our main results. What we see emerging, as in the flat case, is a single shear pattern for a critical width. So if the cells were if the stripe was too narrow, you would not see the flow, because in fact, you have all these terms that penalize splay that would dominate if you had a very narrow stripe. So you would not be able to splay on a short distance. This would be energetically too costly. Here we have a bit of the same thing. If the stripe is too narrow, you don't see flows. Curvature helps if it given that it has the correct sign. Curvature can help to get this single shear pattern. But what we found was quite surprising is that in some parameter regime, in particular if you have friction, you can switch from a non-flowing regime at low curvature and very narrow stripes to directly increasing the activity. You can directly switch to a pattern of flow, which we call the double shear pattern. So on the edges, the cells, they move in a specific direction. And at the tip of the substrate, they can move in the opposite direction. And so it's 50-50. It can be, as a solution, we'll pick one or the other of the pattern.
Jean-Francois, can I ask you something? Sorry, but in these two different models you're showing, the single shear and the double, do you also see a change in the orientation? I mean, because from what you can see on the left side in the single shear, you can see a difference in what you were mentioning before, that in some cases they are oriented perpendicular or in the same line of the curvature, right?
But this is actually exactly, this is what I represent here in blue. This is a cellular orientation.
Yeah.
Okay. What the curvature is doing here is that it favors the cells to be here for this set of parameter, HC negative, the cells want to maximize the experience of the curvature. And so that's why this, in that case, it helps to get the flow. I mean, very schematically. But here you see, because of this splay in orientation of the cells, not individual cell, it's the splay of the cells. This is coherent for an extensile activity. Here we consider an extensile activity. Here you see that the cells, they push on their neighbor, and that's why you get a spontaneous shear flow.
Yeah.
But you could also have this pattern, where the splay has a wavelength which is half of the previous case, where you now have a double shear pattern.
Yeah.
Because here you switch the curvature. So now the cells, they want to go in the opposite. I mean, it's sensitive to the sign of the curvature. The cells, whether they are on top or below the curve, on top or below the curve substrate, they will not have the same behavior. And this has been shown particularly by looking at stress fibers. Yeah. So the sensitivity of epithelial tissue to the sign of the curvature. And this is what we feed in our cross-grain model.
And actually, this one is not including any shear from the external environment, right? Because it's just a shear of the tissue, but there is nothing to do with fluid shear, for example, or something else, because this would change again the pattern. Yeah. Okay.
Exactly. One could consider a shear from the substrate, for example, but this would be an external constraint. Here
or from a fluid on top, you know, you can imagine the blood. Yeah. Okay.
Yeah, exactly. But this would. Yep.
Can I also ask you a question? Because maybe you're gonna discuss this after. If you do, then just skip the question and go to the. But I wanted to ask you, because here you're speaking about shear, which would imply that the most important parameter here is not the shape of the cells, but is their vector field. Velocity field.
Here, but this shear, it's really the relation between the cell shape, the capacity to exert forces on their neighbor, and this setup the flow field that you can see it on this stress equation. I've been a bit I haven't been very rigorous with the signs of my parameters, but it's no big deal. It's just a matter of in essence, if you start having a gradient, think of the gradient of sigma needs to be 0 if you have no friction. So you have a gradient of sigma 0. Here you have the viscosity of your tissue, gradient square, so divergence, Laplace of V, plus gradient of Q. So if you have a gradient of Q, it's going to generate a flow, essentially.
Okay.
So, but I'm not sure I really address your question. But it's really gradients of cell shape that generate gradients of flows, and in particular shear flows.
Right.
I'm not sure I really.
I'm sorry. It could be also the opposite, right? So it would be the
exactly.
Right.
Exactly.
Okay,
the shear flow. That's what I've represented here. But I'm a bit too fast. But the gradient of velocity, you have a flow alignment in your theory, and you can consider different ways. You can consider different signs of the flow alignment. Here, I've really not explicit all of the terms, but here we can assume that indeed, if you have a local shear, it's going to orient the cells. And in fact, that's what fuels the instability. Because you start having a flow, because you start having a shear flow, you also orient the cells.
Right, perfect. Clear.
And that's why you get a second order phase transition. This is my next slide. If you have no curvature, you need to have flows to have cells to be oriented and to acquire a splay. And because you start having flow, you start having splay. Because you start having splay, you start having flow. But this leads to what we call physics a second order phase transition. I mean, the order parameter, I mean, the boundary velocity switches to a non-zero value at a critical activity. However, what we found is that in the presence of curvature, the homogeneous orientation of cells pattern may be perturbed, even in the absence of activity. So because you start having curvature, the cells, even in the absence of activity, could splay because you have curvature. And this leads to something funny, is that in the presence of curvature, you can have flows for whatever infinitesimal activity. You just start to have activity, you start to have flows. And this is something completely different to the paradigm of active matter in confined system, where you need to break, you need to have a sufficiently large activity to start to flow, which is what we represent here in magenta. You need to have a critical activity in order to overcome the elastic constraint from your system. And this value of the threshold, of course, depends on the length of the combine. So what we find is that the curvature induces a first order transition to flow. And physicists are always happy to find first order or second order transition. It's one big element of language. And what we can find also, this is a bit more tricky, but if we play with the curvature, we impose the curvature and we reduce the curvature. That is what we are doing here. We are decreasing the curvature. We find that we don't have the same pattern. For the same activity, you can actually have two patterns of flows. You can either be in a spontaneous shear mode or double shear mode, or in a non-flowing state. So you have metastability, and that's what we observed as emerging from our variation of the curvature. This is what we could call hysteresis or the existence of metastable patterns in our system. And this had not been, to the best of our knowledge, this had not been discussed in confined active nematic or epithelial tissue mechanics. And one thing which is quite funny, I'll skip on that quite quickly, is that we find metastable flow pattern even in the absence of curvature. So here on the left, it's an initial condition. I place my cell along a uniform orientation. I have no flows. I slightly perturb my system, nothing happens, it just relax back to a uniform pattern and no flow. I have a critical activity which is lower than the threshold. Now, I change my initial condition. I am in a regime where everyone was thinking that there would be no flow. But I'm changing my condition. What do I see? My initial condition. What do I see? I have stability of the shear flow pattern. So we have the same set of parameters in those two cases. We just change the initial condition. Same parameter set, we get different patterns. So this, in fact, is not so much surprising. I mean, it's hydrodynamic theory is full of this kind of example of different mode pattern selection. But in that case, in that context, it was not expected to be that striking. And it's quite a challenge for modeling, because if you have the same observable pattern if you have different patterns that can emerge from the same set of parameters, how do you set your parameters in your theory? So here, just based on the cell shape and on the flows, it's not possible in a way to set up the set of parameters. So here, there's lots of things we could explore, fluctuations, etc. I'll skip to my second part. I think I've been so long that I could even stop here, but I'll go very quickly on my second part, and I think I will skip my third part. So I want to talk a bit about these Escher patterns. So what we've been doing with Shaozen Lin, who is currently doing a postdoc in our team, previous things and epithelial fracture were also with him, and with Mark Karna, who is doing his PhD with us. What we've been doing with Shaozen is to develop an active vertex model where cells would exert stresses along the direction of elongation. So this really corresponds to what we discussed before. Cells would exert pressure, would pull on their neighbor in a vertex model with the idea to describe epithelial tissue with an extensile activity. So in vertex model, classically, these were developed to describe bones. There's a term that describes an isotropic pressure within each cell and a term that describes tension along cell-cell interfaces. This Kp is a tension, Ka is related to a bulk pressure. What we're interested in vertex model is to study the dynamics of this plateau border, of these edges between cells, this, which in tissue we call tricellular junction. So in our model, we now include an nematic active stress in the direction of cellular elongation. So like a sigma active, which is beta Q, a bit like what we had in the continuum models. And just doing that was not obvious, and there was a bit of a discussion because we had a different method than in other groups. And I think we have an answer to how this should be done. On the possibility, on the different behavior that would emerge from the choice of forces that would emerge. So here, so you see the set of cells in our vertex model simulation. We are going to increase the cellular activity. So the cells are going to push on their neighbor more and more and more with time, just to illustrate the phenomenology of our system. What do you see? The cells spontaneously flow. But before you started to have flows, you had the emergence of a pattern, which we call the rhombile pattern. And in fact, I'm going to be very fast on this. This rhombile pattern has a finite elasticity. So you have a non-monotonic yield stress as a function of the activity, which is quite surprising. What you see also is that you have flows at a critical activity, which is independent of the system size. You could have taken a huge simulation with 1 billion of cells. The critical activity would have been roughly the same. And this is surprising from a continuum hydrodynamic theory perspective, because what we had before is that you would have flows at a critical confining size. And if your system was infinite, you would always have flows. Okay, so if L was infinite, the flows would start for any tiny activity, any tiny extensile stresses. That's not what we have. There's always a limit. You always need to have an activity that is higher than a critical threshold. And this critical threshold is independent of the size of your domain. And so actually hydrodynamic theories cannot, as such, hydrodynamic theories with a viscosity cannot explain everything. What we have in our system is that we have a yield stress. And this yield stress sets the limit at which, you can take an infinite system, you still won't have flows, even if you have an activity, even at zero activity. And so here's what you get also when you plot the cellular elongation, and you really have these streaming patterns occurring. And well, this starts to really look like an epithelial tissue. You can locally average the cell orientation. You get a pattern of cellular elongation. You can also plot the topological defects in those simulations and represent the stress fields. What do you get? You get, like in experiments, you can describe regions of compression and regions of extension around the topological defect in the flowing regime. The stress, the velocity maps are also coherent with what you have both in hydrodynamic theories and in experiments. And this we've applied to a system of, we've applied to understanding fracture for reasons that I will skip on that part. But what we've shown, which was really not discussed before, is that we can have, using our framework, specifically our framework, we can describe stress fluctuations. And what is correlated to epithelial fractures are, in fact, local, extreme in the local stresses. And that's what triggers the cell, the local epithelial fracture. And so investigating the physics of extreme stresses, I think would be something quite exciting. So here in black, this is simulation, but we have similar plots in experiments. In black are the location of fracture in our simulated tissue. With that, I will very quickly jump to my tracking tree problem. This has been a long collaboration we've had with Thomas Lecuy, where we look at sensory neuron at the surface of the embryo of Drosophila. These structures make really like trees, which are extremely dynamical. And these movies are like, for this early stage of the embryo, are quite a top state of the art. And what we observe is that these branches, these philopodia-like structures, they can grow and retract. But what happens is that when a tree encounters when a branch encounters another branch, it spontaneously retracts. And so the kind of question we addressed is, using an agent-based model as a start, can we describe the resulting morphology? And is this contact inhibition of growth going to impact the global morphology? And what is going to be the emergent tree structure because specifically of this contact-induced depolymerization, this contact-induced retraction? What we are currently doing with a PhD student in the team is to develop a pipeline for automated segmentation and tracking. And we've recently succeeded using also a pipeline that was initially developed for microtubules, if you really want to know the detail. But now we are in a position to really study things which I think are interesting from a statistical point of view and from a physics point of view. Are the tips of these neurons independent? Is the growth something local, or do you have like global correlation? If a tip grows, is it inhibiting every other tips which are connected to the same initial branch? Lots of these questions, I think, are amenable to a physics expertise and to some statistical analysis. And we have a new simulation framework. Can I skip maybe to the end, where we can really recapitulate the structures on a very huge scale. Okay, with that, it's 45 minutes. I would like to thank you, and I would like to thank Jacques Pro, Sam Bell, Benoit, Thomas Lecuy in particular, and also my team members, Xiao Zhenlin, Marc Carnat, and Marc-Éric Perron. And I would like to thank you for your attention. And I hope you're not looking like that right now. Andrea, you're free to laugh.
I am.
Thank you.
Thank you, Jean-Francois. Fantastic.
Okay, maybe I should. This is another picture from the campus. We are 20 minutes away from my building.
Really? That's great. Fantastic.
National Reserve.
Wow, beautiful. Okay, Jean-Francois, maybe I can start with the first question, and then I will stop the YouTube so that to allow also more shy people to ask you things. Okay, so my question is about your continuum model. I was thinking that as you have a curvature in the Z plane for what concerns the cells, you might have an asymmetry between two sheet of two over opposing sheet of layers, which one would represent the basal layer of the cells and the other one the apical layer of the cells.
Totally.
Where you can have different contractility, for instance. We have also seen that there are differences in the contractility, but also the rheological property as a result of that. So
this is a super exciting question.
Sorry, and also the shapes may change, right, between the top and the bottom part of the cells.
So here we are really in a continuum framework. So we are cross-graining on many cells, and we are in a limit where we are not considering we are not considering differently the apical layer from the basal layer.
Right. But I was
So
thinking if you could design.
No, for sure. If we could, this would be super exciting. This would be super exciting. We have we've submitted a paper recently with Tim Saunders on a model where we consider differently the apical and basal layers, and we have a thin shell approach for each of those layers. It would be interesting to. The question is, how do you couple the two?
Yeah, that's a point.
It's no, but there's really a big room for new development. This is super exciting, I think, because
cool.
So this is a bit what we explored in, but not in a curve system, but we performed this vertex model in a 3D context, and typically for this fracture project. And typically what we can get is that you can get compression at the base, but extension at the top.
Right.
And whenever you compress at the base, you in fact extend at the top in some bilateral regime. Some others, no, it's the two are coupled strictly. But no, these are indeed interesting. I mean, in my humble opinion, I think these are extremely interesting questions.